Hello everyone, welcome to ITLS Academy. This is your mentor Dr. Shamaita Basu. So in this session we will be starting with the next topic, that is nucleic acid therapeutic agents. So we will be covering the first part of this topic in this session. Okay, so before starting with nucleic acid therapeutic agents, so what do you basically mean by nucleic acid therapeutic agents? Means where we are utilizing different nucleic acid components or different nucleic acid analogs as a means of therapy to different diseases. Okay, so before that, what are nucleic acids, what are the different types of nucleic acids present? These are the things which I've already discussed before in the previous topics. Okay, so in this session I would just like to recall the concept of central dogma, which is again something I have talked about in details before. Okay, so basically what is central dogma? It is how the information is flowing inside the cell. Okay, information means the genetic information which will ultimately go and code for a protein. So it is always unidirectional, something I've already told, central dogma. So it starts from DNA. From DNA will be formed, that is replication. From DNA, RNA will be formed, that is transcription. And from RNA, ultimately protein will be formed, which is translation. Okay. Now, when the reverse happens, when from RNA DNA is formed, when the reverse transcriptase enzyme is present, which is a special case seen in case of HIV virus, which I will be talking about. Okay, in the next session of this topic, we will be focusing on the different nucleic acid therapeutic agents, and there I will mainly talk about the antiretroviral agents or antiretroviral therapy or ART, which is seen in case of HIV virus or AIDS. Okay. So moving forward, see, now there's a very important role of environment in controlling this central dogma. Environmental factors, genetic factors, and also evolutionary factors are there. So what they're saying over here, organisms respond to their environment via information from sensory input and changes in gene expression. So sensory input means something which is coming from the environment. Something will be which will be the environmental component. Okay, and that will lead to some changes in gene expression. And obviously changes in gene expression means that will ultimately go and affect the central dogma. Okay, and in those cases, see, these path will be changed from RNA to protein, from protein to RNA will start getting formed, from RNA DNA will be start getting formed. Okay, so it's basically a dynamic, immediate, and transient modification of DNA program. DNA program means the central dogma. Okay, dynamic means which is always changing. Immediate means something which is happening then and there. Okay, and transient means again which is something not permanent or not the same. Sometimes it will be there, sometimes it will not be there. Okay, now evolution, the second important factor which will be affecting the central dogma. Okay, the evolution will directly go and affect what? It will directly go and affect the DNA. Okay, so species respond to environment over a long via mutations in the DNA program. So this long time frame is what is evolution. Because evolution is not something which will happen one fine day in the morning. You will just wake up in the morning and you will see that you have grown a tail. No, it's not like that. Okay. So evolution is something which will be happening over a long period of time, at least over 100 years, at least over 70 to 100 years of time it will take for a evolution to occur. So basically it is the response of the of the species to the mutation over a long period of time. Mutation in what? Mutation in the DNA program, means again the central dogma, mainly in the DNA or in the genes. Okay, so evolution is something which is stable, means permanent, it's not something dynamic, and it reflects effect of environment over large time scales. Okay, as we have seen in case of Darwin's evolution, different theories are there, isn't it, that how the giraffes had those long necks. Okay, so it happened over a long period of time, and how? Because of the influence of the environment, because of the effect of environment, some kind of environmental changes had occurred, as a result of which evolution took place. Okay. Okay, so now before starting with the main body or the main content of nucleic acid therapeutic agents, I would just like to give a brief idea about research. Okay, because nucleic acid therapeutic agents are majorly, like a huge proportion of the nucleic acid therapeutic agents are still in the research level. Okay, they have not yet reached the marketing stage or the commercialization stage. Okay, so that's why it's important to have an idea about the different research trials that are going on, particularly concerning the different, you know, medicines or drugs which we will be studying about. So now, what is the basic difference between basic science research and clinical research? So clinical research means where we are utilizing or where we are involving humans as the means of study. Okay, so basic science research is investigation of a subject to increase knowledge and understanding about it. So in the sciences, it usually refers to research performed on cells or animals, knowledge for the sake of knowledge, but can then be translated to other things. Example will be an automatic clock that is critical to satellite based GPS. Just a random example. This example has nothing to do with medical biotechnology. Okay, and now coming to clinical research. So it is any research that involves humans, as I mentioned, and the goal is to improve human health or increase understanding of human biology. And it may be observational, or it can also be interventional. Observational means something which you have applied and then you are observing. Okay, and interventional means something which you have injected or you have intervened with inside the body of that particular human who is the subject of your study. uh, so in case of observational, we observe the different clinical assessments like blood tests, urine tests, okay, sputum tests, like that, spinal fluid imaging, etc., in order to understand more about human biology. An interventional or clinical trial means we determine if our intervention changes a particular condition or a disease. Okay, okay. Now there are some important terms that we need to know when we are talking about clinical research. Okay, so the first one is placebo. What is placebo? It is a substance or thing with no therapeutic effect. So it is basically, placebo effect was first studied in mice, where they were given a set of mice were actually given a therapeutic drug, while the other set of mice were actually not given, like just given a component with no therapeutic effect. Okay. Second is double blind. The researchers and the patients do not know if they are taking placebo or active medication or intervention. Means the patients have no idea what they are being given. Powered means the ability of a study to detect a difference if a difference actually exists. Difference means difference between the two groups or two sets of patients that are being given the same drug or the same therapeutic agent. Okay. Endpoints: an event or outcome that can be measured objectively to determine whether the intervention being studied is beneficial. Means you are determining or measuring something objectively in order to determine whether the intervention that was being used or studied is actually of some use or not. Okay. Okay. Now these are the actually the stages of different clinical trials. So this is again something I mentioned earlier, I think, in the previous topics or sessions. That is, any clinical trial usually has four stages. Okay. Phase 1, phase 2, phase 3, and phase 4. Okay. So basically all these phases are basically studies on humans, where we are gradually increasing the time period and also gradually increasing the sample size. So see, in case of phase 1, we have only 2 humans and it was done for several months. But in case of phase 2, we have taken many humans. Around 5 to 6 humans we have taken. This is just a hypothetical situation. And we continued this phase 2 trial for several months to years. Then in phase 3, we doubled the number of humans. The sample size is doubled or tripled, you can say. And it takes around years to decades, means around 10 to 12 years it takes. And then finally we have the fourth phase or the final phase of clinical trial, which will be ongoing, like it has no end. Okay, and then finally we will go for the marketing or the commercialization part. So this is basically how the process of drug development looks like. So the drug, during the drug discovery phase, means drug discovery means in the lab phase, in vitro phase. Okay, so there we will be studying or testing at around 10,000 compounds. From them we will be coming with our drug. Then we will be going for preclinical testing. Clinical testing means testing on different, like mice, guinea pig, like that. That is preclinical, not on the humans, okay, or not on mammals. Usually, besides humans, we can also go for mammal testing, like we can use chimpanzees or apes, okay. So that will include the preclinical testing, where we will be testing around 250 compounds. Then we will go for clinical testing, where we will be finally testing out, with about 5 drugs, like 5 drugs which we have already finalized, that okay, these are the perfect components for our drug, okay. And then finally we will go with the FDA approval, that is Food and Drugs Association or administration. They will be giving us approval for the one drug which will be showing the maximum number of positive results in the clinical testing part, okay. So see, it will take around till preclinical testing, it will be taking around 4 to 6 hours, sorry, 4 to 6 years. Then the clinical testing part will be taking around 6 to 7 years, and the FDA approval part will be taken around 1 to 2 years, okay. So the total this total phase of drug development will be around 12 to 15 years, which is almost a decade plus an extra 3 to 4 years, okay. Okay, now coming to the main component or the main content of this session, that is the nucleic acid based therapeutic agents, okay. So whenever we are talking about nucleic acid, we know that there are two varieties, DNA and RNA. DNA is deoxyribonucleic acid, which is the more stable form and double helical form, and RNA is ribonucleic acid, which is the less stable form and it is the single stranded form. And the basic difference is that in DNA, we have the ribose sugar, which is deoxy. One oxygen is absent in the fifth carbon position. While in RNA, we do not have any deoxyribose. We simply have the ribose sugar, where the oxygen is present at its designated site. Okay, that is 5 carbon position. So when we will be talking about nucleic acid therapeutic agents, there also we will be having two categories. One will be DNA based and the other one will be RNA based. Okay. So starting with the DNA based therapeutics. So firstly, we will be having the plasmids. What are plasmids? Something I have already talked about, extra chromosomal DNA, which is present only in case of bacteria. Okay. So they are high molecular weight double-stranded DNA, which is abbreviated as dsDNA, containing transgenes which encode specific proteins. Now, what are these transgenes? Transgenes are special genes which are present inside the plasmid and they are responsible for coding specific proteins. Okay. On the molecular level, the plasmid DNA molecules can be considered prodrugs. Prodrugs means just the first stage of a complete drug, that is the prodrug, that upon cellular internalization employ the DNA transcription and translation apparatus in the cell to biosynthesize the therapeutic entity, that is the protein. Means if you apply plasmid or these transgenes which are present on the plasmid as the DNA therapeutic agent or the nucleic acid therapeutic agent, it will just internalize, go inside the cell, and it will activate the transcription and translation machinery. What is it? Ribosome, Golgi body, endoplasmic reticulum. They will just employ it and start synthesizing the therapeutic agent that it is encoding. Okay, so that is the utility behind plasmids. So the mechanism of action of plasmid DNA requires that the plasmid molecules gain access into the nucleus after entering the cytoplasm. So cellular internalization means it is entering into the cytoplasm. But that is not enough. It has to go inside the nucleus for transcription to occur. So nuclear access or lack of nuclear access will eventually control the efficiency of gene expression. In addition to disease treatment, plasmids can be used as DNA vaccines for genetic immunization. Okay, and DNA vaccines are something which are in the forefront of clinical research nowadays. Scientists are trying to come up with DNA vaccines for various diseases like HIV, malaria, etc. Okay, so that is also that also will be an interesting avenue for plasmids as nucleic acid therapeutic agents. And the first federally approved human gene therapy protocol was initiated in 1990 for the treatment of adenosine deaminase deficiency, ADA deficiency, and in 2002 the successful gene therapy curve for SCID was reported. What is SCID? Full form of SCID stands for Severe Combined Immunodeficiency Syndrome. Okay. So SCID was the first disease where this gene therapy was first implemented successfully. Gene therapy is again something we will be talking about in details in the upcoming sessions. Okay. And in 2003, the Chinese Drug Regulatory Agency approved the first gene therapy product for head and neck squamous carcinoma under the trade name Gendicin. And currently, there are diseases with complex etiologies such as cancer and neurodegenerative diseases such as Parkinson's and Alzheimer's disease, which are being targeted using these plasmids as DNA therapeutic agents. And we also have DNA vaccines which have been used to prevent allergic responses. Okay. Okay, now the second category which will be coming under DNA-based therapeutics, will be the oligonucleotides for antisense and antigen applications. So what are these oligonucleotides? These are short single-stranded segments of DNA that upon internalization can selectively inhibit the expression of a single protein. So it is basically working on the strategy of inhibiting the expression of a single protein which may be harmful or which may be the result of that particular disease. Okay. So for antisense applications, oligonucleotides interact and form a duplex with mRNA and inhibit its translation or processing, consequently inhibiting the protein biosynthesis. So basically the concept is that since we are trying to inhibit the protein production and the protein will be forming from the RNA, that is the mRNA. So if we can go, if that oligonucleotide can go and form a complex, a duplex with that mRNA and inhibit the mRNA, then automatically the protein biosynthesis will be stopped, isn't it? So that is how this oligonucleotides are working. For antigen applications, oligonucleotides must enter the cell nucleus, form a triplex with the double-stranded genomic DNA, thus inhibit the translation as well as transcription process of the protein. So instead of binding or forming the duplex with the mRNA, it can directly go and form the triplex with the double-stranded genomic DNA. Why triplex? Because the DNA is double-stranded. So already two strands are there. Now the oligonucleotide is going and forming the complex. So ultimately the complex which will be formed will be the triplex, isn't it? So if the triplex is formed with the genomic DNA, then both transcription as well as translation will be inhibited. For therapeutic purposes, oligonucleotides can be used to selectively block the expression of proteins that are implicated in diseases. With successful antisense inhibition of proteins in animal models, the first antisense drug, that is fomivicin sodium, was approved for the treatment of cytomegalovirus retinitis in AIDS in 1998. So cytomegalovirus is basically a virus which causes additional infection in those patients who are basically suffering from AIDS, because AIDS means the disease caused by HIV virus where the immune system is totally broken down. So if the immune system is broken down, then that person automatically becomes susceptible to various other infections, which will include other viral infections also. And one such other viral infection is cytomegalovirus, whose infection is very commonly seen in AIDS patients, and it mainly causes retinitis. It affects the retina of the eye, inflammation of the retina of the eye. And for this particular condition, cytovirus retinitis, this particular antisense drug was used for the first time successfully in 1998. That is fomivicin sodium. The name is important. Please try to remember the name. I will just highlight it for you. Okay. Antisense oligonucleotides such as MG98 and ISIS 5132 are in clinical trials of cancer. Also used against hepatitis C virus, human cytomegalovirus, which is also abbreviated as CMV virus, human immunodeficiency virus, that is HIV virus, and papilloma virus, that is HPV. Okay, now moving forward, the third category of DNA-based therapeutics after plasmids and oligonucleotides will be DNA aptamers. Okay, now what are these DNA aptamers? They are double-stranded nucleic acid segments that can directly interact with the proteins. Aptamers will interfere with molecular functions of disease-implicated proteins or those that participate in the transcription or translation processes. Preferred over antibodies in protein inhibition owing to their specificity, non-immunogenicity, and stability of pharmaceutical formulation. So they are more preferred over antibodies. Why? Because they are having a non-immunogenic property. So they will not be eliciting any immune response which is unwanted. Okay. And the fourth category will be the DNA enzymes. What are they? DNA enzymes are analogs of ribosomes with greater biological activity. RNA backbone chemistry is replaced by DNA motifs that confer improved biological stability. Because, as I mentioned, RNAs are less stable; they are comparatively unstable than DNA, isn't it? So, if we can use the DNA enzymes as analogs of these ribozymes, then we will be, like doing away with the instability of the RNA or the ribozymes. Okay. So DNA enzyme directed against vascular endothelial growth factor receptor 2 was confirmed to be capable of tumor suppression by blocking angiogenesis upon intratumoral injections in mice. Okay, so intratumoral injection means inside the tumor, inside the tumorous growth, the tumorous mass. This injection was given. What injection? This DNA enzyme injection, and it was found to suppress the tumor by blocking angiogenesis. What is angiogenesis? Angiogenesis is formation of new blood vessels. Okay, genesis means formation and angio means new blood vessels, which is seen in case of tumors, particularly cancerous tumors. Okay, so if we can utilize this DNA enzymes to inhibit the process of this angiogenesis, then automatically the tumors will be suppressed, and it is also used against cardiovascular diseases and cancer. Now coming to, so we are done with the DNA based therapeutics. Now we will be starting with the RNA based therapeutics. So the first category that will come under RNA based therapeutics will be the RNA aptamers. So just like DNA aptamers, we also have RNA aptamers. So these are obviously RNA, so they will obviously be single stranded nucleic acid segments that can directly interact with proteins. Recognize their targets on the base of shape complementarity. Their binding specificity and affinity for target are extremely high and similar to the MABs. MABs means monoclonal antibodies. And they are used against HIV-1 transcriptase. Transcriptase means the reverse transcriptase enzyme, which is a characteristic of HIV virus. Clinical study on humans with injection of anti-VEGF aptamers in eye showed that 80% of the patients improved the eyesight, and not only that, they showed no side effects. What does VEGF stands for? Vascular endothelial growth factor. Okay. Okay, so coming to the second category of RNA-based therapeutics, that is RNA decoys. What are they? They are designed to provide alternate competing binding sites for proteins that act as translational activators or mRNA stabilizing elements, prevent translation or induce instability and ultimately destruction of the mRNA. Overexpressed short RNA molecules corresponding to critical cis-acting regulatory elements can be used as decoys for trans-activating proteins. Okay, now there are some advantages of these RNA based therapeutics above or over other nucleic acid based strategies, means mainly the DNA based strategies. What are they? Decoys are less likely to be affected by variability of the infectious agents, because any mutation in the trans activating proteins affects not only binding of decoys but also binding to the endogenous targets. And the decoys can also be used against HIV infected cell, which is a huge plus point. Okay. Now coming to the third category of RNA based therapeutics, that is antisense RNA. What are these antisense RNA drugs? They are short stretches of deoxyribonucleotide analogs that bind to specific complementary areas of mRNA by Watson-Crick base pairing to block the gene expression in a sequence-specific fashion. So basically, they are complementarily binding to those areas of mRNA which will be following the Watson-Crick base pairing, means ATGC base pairing, to block gene expression in a sequence-specific fashion. AD means antisense drugs may induce an RNase H which cleaves the RNase H means an hydrolyzing enzyme which hydrolyzes RNA, which cleaves the mRNA at the site binding. Or physically, it can block the translation or other steps in mRNA processing and transport to protein biosynthesis. AD work at an early stage in the production of a disease-causing protein. It can be applied to a number of diseases where the basic pathophysiology involves an overexpression of a given protein molecule. So basically, in any disease where a particular overexpression of a particular protein is the main cause of that disease, in those diseases we can use ADs, antisense drugs. Okay. Antisense odins can base pair with the gene's transcript and constitute a new technology for the control of gene expression in prokaryotes and eukaryotes, including the mammalian cells. So this is something which is under research. And newer antisense oligonucleotides may offer improved pharmacokinetic and safety profiles because of reduced non-specific interactions. Now, there are some advantages as well as there are some disadvantages of antisense RNA. What are they? Disadvantage will be high expression required to successfully bind to all the target RNA. And advantage will be lack of immunogenicity of antisense constructs such that the oligonucleotides and cells producing them will not be destroyed by the host immune response. This is again something important which I already talked about in case of when I talked about the DNA based therapeutics also. That non-immunogenicity will be a boon. It will be a plus point. Why? Because otherwise the host immune response will be elicited and that will lead to destruction of the therapeutic agent. Okay. Antisense RNA alone is not potent enough to produce complete inhibition in vivo. In vivo means in real situation inside the body. Enzymatic moiety can be induced with antisense oligonucleotide, which will cleave the target RNA as well as the RNA duplex that has been formed. So alone, antisense RNA is not enough. We need an enzymatic moiety which will be acting together with the antisense RNA to exert the function. Okay, and these enzymatic RNA strands will be known as the ribozymes. Okay, now coming to the next category of RNA based therapeutics, again very important, that is the small interfering RNAs or the RNAi. Okay, the phenomena exerted by small interfering RNAs is known as RNAi. The small interfering RNAs are abbreviated as siRNAs. Okay, S for small, I for interfering. So RNA interference, it is a post-transcriptional mechanism of gene silencing through chromatin remodeling, inhibition of protein translation, or direct mRNA degradation, which is ubiquitous in eukaryotic cells. Means it's a common phenomena. It happens everywhere in the eukaryotic cells. Now, the small interfering RNAs or the siRNAs, it can be used for down regulation of disease causing genes through RNA interference. Down regulation means basically inhibiting those genes which are responsible for causing the disease. Short double-stranded RNA segments with 21 to 23 nucleotides and are complementary to the mRNA sequence of the protein whose transcription is to be blocked. So this is basically the definition of the small interfering RNA or the siRNA. So they are having a length of around 21 to 23 nucleotides in length and they are complementary to the mRNA sequence of the protein which needs to be blocked because overexpression of that protein is the trouble which is causing the disease. So we need to block the expression or the synthesis of that protein. So in turn, we need to block the mRNA, and that's why this siRNA will simply go and form a complex or duplex with the mRNA and thereby inhibit the production of the protein. Okay. On administration, the siRNA molecules will be incorporated into the RNA-inducing silencing complex, which is also abbreviated as RISC, which bind to mRNA of interest. So now this RISC will go and bind to the mRNA of interest and stimulate mRNA degradation mechanisms such as nuclease activity, which leads to silencing of the particular gene. Introduction of foreign double-stranded RNA can initiate a potent cascade of sequence-specific degradation of endogenous mRNAs that bear homology to the double-stranded RNA trigger. When double-stranded DNAs are introduced into the cytoplasm, they are processed by the RNase 3 enzyme, which is called dicer. And this dicer will go and cleave the long double-stranded DNA into short 21 to 23 nucleotide duplexes, which have symmetric 2 to 3 nucleotide 3' overhangs and 5' phosphate and 3' hydroxyl groups. As few as 7 contagious complementary base pairs can direct with the this RNAi mediated silencing, and it is used to inhibit HIV, hepatitis and influenza infections. Since siRNAs do not integrate into genome and they offer greater safety than plasmid molecules, it is possible to deliver a cocktail of siRNAs targeting multiple disease-causing genes in a single delivery system to inhibit the complex diseases such as cancer where several genes are malfunctioning. And then we have last category of RNA based therapeutics. We have microRNA, which is again abbreviated as miRNA. So these are a class of naturally occurring small non-coding RNA molecules with 21 to 25 nucleotides in length. These molecules are partially complementary to the mRNA molecules, and their main function is downregulation of gene expression via translational repression, mRNA cleavage, and deadenylation. So instead of going and forming complex with the mRNA, they will just cause deadenylation of the poly A tail, and also they will cause cleavage of the mRNA. So instead of mRNA degradation, they are going for cleavage and deadenylation. They were first described in 1993, and the term microRNA was coined in 2001. Central online repository for microRNA nomenclature, sequence data annotation, and target prediction is MIR base, which is hosted by the Sanger Institute. MicroRNAs are transcribed by RNA polymerase II called pri-miRNAs that compete with the 5' cap and the poly A tail. In the nucleus, the pri-mRNAs are processed into pre-miRNAs by the microprocessor complex, which consists of the RNase 3 enzyme DROSHA and the double-stranded RNA POSHA, which is also known as DGCR8. These pre-miRNAs are exported by the karyophorine exportin and the RANGT. These are just exporting complexes or names of different protein complexes, not that much important. So they will be additionally processed by the RNase 3 enzyme dicer, which is also responsible for the initiation formation of RISC, which is responsible for the gene silencing observed due to miRNA expression and RNA interference. Okay, so I think that was an overview about the two main categories of nucleic acid therapeutic agents that can be used, that are in used. And in the next session of this topic, the second session of this topic, we will be talking about in details about the different nucleic acid therapeutic agents used in ART, mainly for AIDS treatment. Okay, so till then, thank you, goodbye. Please like, share and subscribe ITLS Academy.